to talk about photosynthesis today. And part of this it is within cellular energy. So we've already talked about cellular respiration. Today we're going to be talking about photosynthesis within the light and the dark reactions today. So within photosynthesis, we're going to be able to answer these three questions. What are the two phases of photosynthesis? What is the function of the chloroplast during light reactions? How does electron transport chain be described and diagrammed? So the first thing we have to understand is that with photosynthesis, this is going to be through organisms that are autotrophs, where they make their own organic compounds. And so in this case, our organic compound that we are looking at specifically is glucose, or C6H12O6. This is going to be done through this process of photosynthesis. So we're going to look at the overall equation of photosynthesis. We have 6CO2 plus 6H2O is going to yield, and so my carbon dioxide and hydrogen, or my water, are my reactants, while my glucose and oxygen are my products here. Now, we see the same thing, the same equation, but flipped within cellular respiration. So remember, they are inversely related. So photosynthesis occurs in two phases. We have the light-dependent, or light reactions, and our whole goal is to absorb light energy and make it into chemical energy, or in this case, create bonds through the terms of ATP and NADPH. While in the dark reactions, or what we also call the light-independent reactions, or what we also call the Calvin cycle, what we are doing is the energy from the light-dependent is going to make, we're going to take the chemical energy that we made through the use of ATP and NADPH to make glucose molecules. So chlorophyll is a big key in this, and what it's going to do is this is a molecule that absorbs light energy, and so in plants, chlorophyll are found in the organelle found only in plants called the chloroplast. And so within the chlorophyll, we have pigments. So we have chlorophyll in chloroplast, and we have pigments in chlorophyll. And so its whole goal is a compound that absorbs light energy, and so what the colors that you see are what's reflected from the light energy that it's not absorbing. So we have chlorophyll A and chlorophyll B are main players. Usually it's mostly chlorophyll A, and the second most chlorophyll B, and the third is what we're going to talk about next. And so these are the most common types, and they're going to absorb different amounts of light or different colors of light. So our third one, that's the third most within these plants. And we have 6 different types of these. I'm not going to ask you the 6 different types, but just know that there are different types of carotenoids. And these are accessory pigments as well. And what they're going to do is they're going to absorb light that neither chlorophyll A or chlorophyll B can absorb. And this increases the range of light that the plant can absorb to increase the amount of photosynthesis a plant can go through. And so within these carotenoids, there's also non-photosynthetic parts, it's like within these fruits. And they're going to carry more carotenoids than chlorophyll, and they have a variety of colors as well, as we see here in this picture. In the fall, leaves are going to lose their chlorophyll, and the leaves are going to take the colors of the carotenoids and present in them. And so we see these light here. And from here we have carotenoids within this leaf, and so the chlorophyll A and chlorophyll B, they're going to leave, and so we have carotenoids that are only going to be visible. So our first stage of photosynthesis is going to capture light energy and it's going to transfer this energy, and this is going to take place in the thylakoid membrane. And so we see here this little green disc, that is my thylakoid, and what's going to happen is we're going to include groups of molecules called photosystems that we're going to look at here in just a minute. So this is going to be our part where we bring in water or sunlight. We're going to bring it to the thylakoid, and then we're going to make some molecules here. So our first photosystem that we're looking at is called photosystem 2, and it's going to happen first. So it's going to capture and transfer energy. So chlorophyll is going to absorb the energy from the sunlight. It's going to energize electrons from the chlorophyll, which is then going to enter into the ETC, or the electron transport chain that we saw in cellular respiration. Water molecules are going to split through a process called hydrolysis. So hydro, water, lysis, to split or break apart, so water splitting. Oxygen is going to not be used, so we're going to release it as waste, which is what we breathe in. And then electrons are going to replace the electrons lost excuse me, into the electron transport chain. So we broke water, electrons from the hydrogen are going to replace it in the photosystem, oxygen is going to be released, and then hydrogen is going to be transported across the thallic membrane. And so without this process, light reactions would not occur because there's no hydrogen for that gradient that we're going to see later. And so, again, step 1, energy absorbed from the sunlight. Step 2, water molecules are broken down. Step 3, hydrogen ions are transported across the thallic membrane. So, our light-dependent reaction, our whole goal is to produce ATP and NADPH. And so, within this, our hydrogen ions move through this channel in the thallic membrane. We have this ATP synthase that attaches to the channel of this thallic membrane, and it makes ATP. Using energy from the hydrogen gradient to make ATP is known as what we call chemoosmosis. Now, chemo is to make a chemical, osmosis, movement of water. Remember, we broke down water to move the hydrogen ions. And so this is a picture of the ATP synthase that we'll see after photosystem 1. So, photosystem 1 is going to capture energy, another wavelength of sunlight, and produce energy-carrying molecules in the form of NADPH and ATP. So chlorophyll is going to absorb the energy from the sunlight. It's going to energize the electrons used to make NADPH. The electrons from the electron transport chain of photosystem 2 replace the ones lost in photosystem 1. NADPH is transferred to the light-independent reactions. And we're also going to be making ATP as hydrogens flow through the ATP synthase here to make ATP to also go to the light-independent reactions. So we see here energy is absorbed by the sunlight. Electrons gain energy, and they're going to drop down and be lost into this space here, right here. It's going to join up with hydrogen to form NADPH with the 2 electrons that we gave from the 2 hydrogens earlier. So here's our input and output, the overview of what occurs here. So we have input is the sunlight in photosystem 2 and in photosystem 1. Molecules go in as in water, and this is the hydrolysis. We're going to split this in photosystem 2. Hydrogen crosses the membrane. Electrons go to photosystem 2 to replace the electrons lost through the electron transport chain. And oxygen is left. It's what we breathe in. So then we have the products, ATP, NADPH, made in photosystem 1, and oxygen is released again in photosystem 2. So now, after we have the light reactions in photosystem 2 and photosystem 1, the dark reactions are going to occur, and this is where we're going to use energy from the light reactions to produce the sugar. So this is going to occur in the stroma, or what you guys might call the cytoplasm of the chloroplast. And so what we're going to do is we're going to use what we made in light reactions and use them in the dark reactions. Unlike cellular respiration, we had to move stuff around to try to figure out where it went. In photosynthesis, it's literally light reactions to dark reactions. The only thing that's not going in and out, and we're going from one place to another, is carbon dioxide and water, or and oxygen, which is what we'll look at here in a minute. So the light-independent reaction, or the dark reactions, or also known as the Calvin cycle, make sugar, or in the term glucose. And so from here, it's going to take place in the stroma, like we talked about. It's going to require an input of carbon dioxide, 6 of them from the atmosphere, and it uses the energy to build a sugar in a cycle of chemical reactions. So the Calvin cycle is a series of enzymes, controlled reactions that produce a 3-carbon sugar from CO2, also known as G3P. Now, this process is known as carbon fixation, and the carbon is bonded into organic molecules. So, I like to start with the end on what's left over so we know where we go. So, this is going from kind of what we do with the Krebs cycle in cellular respiration. Similar process here in the Calvin cycle in photosynthesis. So we have this ribulose 1,5 biphosphate here that's what's left over. And what it is 2 G3Ps that we added 2 phosphates to in order to get ribulose 1,5 biphosphate. And so that's going to be very important that we have 2 G3Ps that are recycled. So we have carbon dioxide and hydrogen and water coming in to this. And so what we're going to do is we're going to bind these to the ribulose 1,5 biphosphate. But we need help. And we need help by an enzyme called ribisco. And so ribisco is going to come in like what coenzyme A did to form acetyl-CoA. And so we're going to come in, we're going to bind, or we're going to fix carbon to these two molecules. And so what's going to happen is we're going to bring in ATP and NADPH into this reaction through a series of enzymes to reduce the charge. So we're giving away hydrogens, so it's becoming more negative, so we're reducing the charge, and we're putting in some phosphates here. And this is in order to make what we need to make in terms of G3P. So we're going to make G3P, and on this turn, so we're bringing 3 carbon dioxides, we're making 1 G3P. So when we do this, we have to get back to where we were with ribulose 1,5 biphosphate. In order to do that, it was made up of 2 G3Ps. We started out with 2 G3Ps mixed with some phosphate. Then we added another G3P. So we're going to recycle the 2 G3Ps, and we're going to add phosphate to them, or what we call regeneration, to make ribulose 1,5 biphosphate. Then we're going to save 1 G3P to bind it with another one through another cycle in order to make glucose. So we went through one cycle. Here we made 1 G3P, we recycled 2. Now we have to bring in another 3 carbon dioxides and some more water to go through the whole cycle again to make 1 more G3P to combine to make glucose, and recycle 2 more in order for it to reoccur. So the end result is we're making 6 G3Ps. 2 are being used to combine to make glucose, because there are 2 3-carbon sugars. Glucose is a 6-carbon sugar. And we're recycling the other 2 every time, or in this case, since we went through 2 cycles, we're recycling 4 to have the process start over again. So again, our input is ATP and NADPH, as well as carbon dioxide. Our product is glucose in the form of 2 G3Ps. So the equation for overall photosynthesis is here, and this is a picture that you may or may not see again, so I would suggest that you look at it and be able to understand what you can. This is an overview of what's photosynthesis and how they work together. This RUBP that's here is the ribisco biphosphate or ribulose biphosphate. This is the G3P that we're going to combine to make sugar or glucose. This is my photosystem 2, photosystem 1 here. And then here's another picture for you, just to kind of give you another little picture as well. And then you should be able to answer this question at the end. And that is photosynthesis. So make sure you're able to look at those things and be able to understand what occurs within these processes. And hope you have a great day. And if you have any questions, please let me know.